thank you, and thank you, Kat, for your presentation.
Thank you for having me.
Okay, right, and now we move on to Ethan Barnes from Greenlight. Ethan, if you could switch on and get your presentation ready. That's it, great, very sharp. Okay, so welcome, Ethan. It's by contrast with Australia, it's early morning in USA, and in fact we made him wake up even earlier because we wanted to just check that we had a good contact with him. So thanks for joining us so early in the morning, Ethan. Ethan is from Greenlight Biotechnology, who've been working on developing a number of products, RNAi products. Today, he's going to talk about the particular dsRNA insecticide that they have taken quite a long way down the road towards development and production. So I hand over to you, Ethan, and we look forward to your presentation.
Thank you. Well, yes, thanks Jeremy and Bruno for organizing this and giving us an opportunity to talk a little bit about Ledprona, which is Greenlight Bioscience's very first product. And it's a foliar applied RNA bioinsecticide that selectively controls Colorado potato beetle. See if I can get it to advance here. So the information I'm be presenting today is property of Greenlight Biosciences and shouldn't be copied or distributed without written permission. Ledprona, or GS2 as I'll be referring to it throughout this talk, is expected to be the first sprayable RNA product registered in the USA. So that's a huge milestone for RNA technology. Greenlight Biosciences has conducted 3 years of field trials on potatoes, and we placed those field trials across the major potato producing regions of the US, Canada, and EU. Colorado potato beetle, or CPB, can be very devastating to potato plants in these regions. And Ledprona offers very good protection against Colorado potato beetle. Its performance is similar to current industry standards on the market, and it's on track to becoming a new commercially available solution in 2022-23. So Ledprona is an aqueous-based formulated product that meets all requirements and expectations of a formulated agricultural product. Notably, it's stable in long-term storage conditions or in normal transportation conditions. It's also easy and safe to handle by the end user, and it has no negative impact on the biological performance of the active ingredient, the RNA. So Ledprona's development has been driven by the understanding of the product itself and how Ledprona can be positioned into a production system. So the initial development focused around sequence selection, selecting the correct sequence, and determining the effective dose on Colorado potato beetle. Development then transitioned into looking at Ledprona in the field, so in IPM programs and with mixture partners. We then defined the use rate and the appropriate application interval that Ledprona should be applied in the field. And we also tested Ledprona in real application conditions. We had some field trials in both Europe and Canada, and we're excited about the possibility of expanding Ledprona into those geographies. So Ledprona is designed to specifically only target Colorado potato beetle and no other insect species, including beneficial insect populations. So if we look at the field trials, in general, our field trials have been with 2 to 4 row potato plots with 4 replications. They've all been sprayed with small plot equipment with normal application conditions for what producers would be using in potato production. The initial foliar application of Ledprona has been targeted for 50% CPB egg hatch. So that's the point when we still have 50% eggs in the field, and the other 50% of the eggs have hatched and are now L1 or first instar larvae. As far as data collected, we've had all trial locations collect insect counts by life stage all the way through adulthood. We've had them collect whole plot defoliation and phytotoxicity as well. Although it should be noted that over the last 3 years of conducting field trials, Ledprona has never had any phytotoxicity reported. We've also collected yield on a number of the field trials. So Ledprona performs similar to industry standards. We see comparable efficacy with Ledprona and current industry standards in the marketplace. So in this first graph, we have the number of larvae per plant here. And what we're looking at is two different rates of Ledprona or GS2. We have a 0.5x rate in brown and a 0.25x rate in gray. So by day 14, this is 14 days after the application of Ledprona, and this is a greenhouse study. We see complete mortality and have no living larvae left on the plants. And that's similar to the industry standard, in this case, standard number 2, which is the orange bar here. Ledprona works a little bit differently than most industry standards. What we typically see is that insects, the larvae, stop feeding around 1 day after application. And then we start to see mortality around day 4 through day 10. These photos here kind of portray what we see in the field as well. This is the untreated check at 14 days after application, where you see very healthy, growing larvae that are clearly feeding well. These larvae here are treated with GS2, so these larvae clearly are not growing. They stop feeding again around day 1, and then they turn moribund until they die around day 4 to day 10. They often turn this black in color as they're just sitting there, no longer defoliating the plant. The bars here depict defoliation across the plants in the greenhouse study around day 14. So 60% defoliation in the untreated check, and the two GS2 rates, Ledprona rates down here, are below 5% and similar to this industry standard number 2 again. Also want to show you what it looks like in the field. In the field, we're seeing similar protection from Ledprona against defoliation. So here we have 6 trial locations summarized across the 2019 field trial program, and we're looking at the percent protection and defoliation. So of course, the untreated check offers no protection against defoliation, as there's no pesticides applied in those plots. And typically what we're seeing is that a 0.5x to 1x rate of Ledprona is providing similar protection against defoliation compared to industry standards. In these field trials, we had 2 industry standards, standard number 1 and standard number 3 here. So just to show you kind of what that looks like in the field when we're seeing it in action, we have Ledprona at a 1x rate here on the left compared to standard number 3, and you're seeing very comparable amounts of protection, pretty much complete protection out of both of those products. And Ledprona compared to standard number 1. Again, you can't tell a difference between the 2 plots. We have the untreated check in this first photo just next to standard number 3, and you can see the amount of defoliation happening. At this trial location, defoliation got much worse in the untreated check in the coming weeks. However, Ledprona in both the standards held strong over the next few weeks. It's also important to note that the use rate of Ledprona is actually much lower than most current industry standards in the marketplace. So Ledprona also needs to be able to fit into current grower programs. So a lot of our development efforts at Greenlight have focused around making sure Ledprona can fit into current industry practices. So Ledprona works well on both the first and second generation. And here I can walk you through some of the data of Ledprona in a growers program. We have defoliation during the second generation of Colorado potato beetle. So most of our trial locations are experiencing 2 generations of Colorado potato beetle. The untreated check here is first, where we see around 80% defoliation. So it's a high defoliation trial location that we're showing. And the next treatment is the industry standard program. So we have a neonic down in furrow at planting. And the first generation is controlled by 3 applications of standard number 2, and the second generation is controlled by 2 applications of standard number 4. The next 2 treatments are the same neonic down at planting, but now we have Ledprona for controlling either the first or the second generation of Colorado potato beetle. And what we see is very low defoliation from both of those programs, whether Ledprona is in the first generation or the second generation. The last two treatments then are Ledprona in the first or second generation again, but in this case, we do not have the neonic down in furrow at planting. And then we have standard number 2 in either in the opposite generation with Ledprona. So again, we see very low defoliation, under 20% defoliation without the neonic down at planting. But it's important to point out at this location that they had severe leafhopper feeding during the second CPB generation. So we see a little bit increased defoliation here, which is mostly due to the leafhopper feeding. Standard number 2, which would have been in the second generation of this orange treatment, does not have much activity on leafhopper. And of course, GS2 or Ledprona is designed to specifically only control Colorado potato beetle. And if we consider the neonic treatments, neonic obviously offers some protection against leafhoppers, and standard number 4, which was applied in the second generation of the standard program, offers good leafhopper protection. So I want to show you the exact same treatments, but now we're looking at the yield at the end of the year. So here we're looking at the total marketable yield. Of course, the untreated check has the lowest total marketable yield. And as we saw on the previous slide, there's about 80% defoliation, so we would expect that to have quite an impact. The best yielding treatment then was the industry standard program, again, offering the most protection against the leafhoppers, but also having really good control of the Colorado potato beetle. GS2, following a neonic either as the first generation or second generation, had similar yield compared to this industry standard program. And then the last two treatments still yielded much higher than the untreated check, but without that neonic down at planting and no activity on leafhoppers in the second generation, we see a little bit lower yield, but still much higher yield than the untreated check. So it's important, as I mentioned before, that it fits into current industry practices and to current grower programs. So Ledprona needs to be able to last for at least 7 days in the field to be competitive in the marketplace. So here we're testing different application intervals. First, we have the 7-day application interval in blue. So that's 7 days in between treatments. We have a 10-day application interval in red and a 14-day application interval in green. In this untreated check, we see 40% defoliation. So that's across 6 different trial locations in the 2020 program, so this summer. And what we typically are seeing is that 7 to 14-day application intervals with Ledprona is providing considerable reduction in defoliation. However, when we look at the 7 to 10-day interval, again, this blue and red bar with Ledprona or GS2, we see a little bit more consistent control and protection from Ledprona. And if we look at the standard number 2 here, we have them on the same application interval, and we see that that 7 to 10-day application interval is very competitive with standard number 2. We also tested Ledprona with an adjuvant. So in some trial locations, you can see a slight improvement when you add certain adjuvants to Ledprona, but it's certainly not always the case and isn't always needed. So especially across the US geographies, it's important that Ledprona can be applied in different application systems. So growers across the US are applying by different mechanisms. So with tractor mounted or ground application vehicles, they're typically applying somewhere around 200 liters per hectare. So we have Ledprona at a 0.5x rate and 200 liters per hectare, and we see no defoliation in those plots. And we see that the untreated check here has pretty high defoliation. Growers are also applying through airplane. So applying pesticides to potatoes through airplane, which is around 50 liters per hectare. So here we're applying at 50 liters per hectare to simulate what an aerial application would look like. So fairly low application volume. And we see really good protection again. And it's comparable to the standard practice. The final pane here, we're applying the same rate of Ledprona, but in nearly 36,000 liters per hectare. So an incredibly high amount of water applied with the Ledprona, and that's to simulate chemigation, so pesticide applied through an irrigation system, where you would be applying mostly water, but also injecting in the pesticide at the same time. Again, we're seeing really good protection, and it's much better than this untreated check here when we're applying via chemigation. So at Greenlight Biosciences, we're really proud of the progress made with Ledprona, and we know it's well on track to becoming a commercial product in the next few years. And really what I would like you to take away from today's talk is that Ledprona works well in various growers program. So it's fairly flexible in whether it's applied in the first or second generation. We've also determined the commercial use rate that should be used, which again is much lower than current industry standards in the marketplace. We've determined that the application volume should be around 7 to 10 days, which is very competitive with current industry practices. We also know that additions of certain tank mix adjuvants can improve performance slightly, but it's certainly not needed in all situations. It also works well in different application methods. As you've seen in the last slide, we can apply it anywhere from 5 gallons per acre all the way up to nearly 4,000 gallons per acre. So it's fairly flexible in how you apply it and the method that's applied in. And we've seen no issues with tank mixing with other insecticides, fungicides, or oils. We've seen no tank mix compatibility issues and no decreased efficacy from Ledprona when tank mixing it. Again, we're really excited about expanding into the other geographies, into Europe and Canada, and we've had really good results from field trials in both of those locations. So we're excited about that potential of expanding into those geographies with Ledprona. I think we're going to hold questions until the end, but again, we're really excited about the progress made with Ledprona, and I'd be happy to answer any questions I can at the end of the webinar. Thank you.
Thank you very much, Ethan. Very interesting talk. There are perhaps 1 or 2 specific questions that you might like to address now, and
Sure.
then more general ones which we could pick up at the end. I don't know how much you can reveal on the on this thing, but there is people wanted to know more about the formulation and how much the efficacy has been affected by different formulations.
Yes, that's a good question. Yeah, there's not too much I can reveal about the formulation. The efficacy prior to formulating the product was good as well, and our focus for the formulation was just to stabilize it in storage and transportation, as well as just making it a product that can be used like any other pesticide on the marketplace.
Okay, and what can you tell us about the target gene, and were there different effects on different life stages of the beetle?
Yeah, so I can't reveal the target gene specifically, but Ledprona does work on larvae and adults, so it does not have effect on eggs. Ledprona needs to be ingested by the larvae or adult beetles.
Okay, thanks. And can you tell us what the application rates of the actual double-stranded RNA were? Because you gave the overall Ledprona rates, but what about the actual application rate of the double-stranded RNA?
Yeah, so we're not going to be sharing the rates just yet. Those will probably be coming in the next few, or over the next year or so. All I can say is that it is much lower than most industry standards, so you can expect to see a low rate of the actual dsRNA once we reveal that.
Okay, thanks very much, Ethan. Yes, please stay with us because there are some other more general issues about pesticides and registration and so on, which we can perhaps take at the end. So
Thank you.
Thanks very much for your talk. Very interesting. Now we move on to a talk by Salvatore Appaia from RNAi in Italy. And he's going to give us a general introduction to some of the biosafety issues associated with topical applications of double-stranded RNA. Start from the starting point of perhaps what we know about it from other studies that have been done from in-planter studies and so on. So Salvatore, are you ready to address us? And do you have your presentation ready?
Yeah, thank you very much, Jeremy. Thank you for giving me the chance of closing this first seminar. And good afternoon to all colleagues. Good morning to those who are listening from the Americas. And I don't know if Nina is still with us or if she's, good night to Nina.
I'm still there and enjoying the talks. Thank you.
Okay,
Good.
glad. As Jeremy said, I work for RNAi, which is the Italian national agency in charge of applied research in several fields. And what you can see here is our headquarters. Some colleagues in Atlanta have been there. Not a particular nice building in Rome, we have much better, but a few meters from the Tevere River, so it's not a bad part of the city. The issue of biosafety, as Jeremy already mentioned, will be dealt in depth in next webinar. However, it's important that we have this, I cannot really change slides, apparently. Let me try again. Okay. I want to clear up immediately. That's why we wanted to have this first introduction to biosafety issue. I want to clear up a misconception that biosafety might represent a burden to development of new technologies or products. Indeed, what I and the working group of iPlanta working on this aspect, we are considering environmental safety framework in the sense that for environmental safety framework, we need protocols, scientifically developed protocols, which can become practices and hopefully policies. All this framework is to ensure that there is a safety and well-being of any organisms, including humans. I will be talking about the environment today, but of course the food safety is another big important issue. And all this is based on scientifically sound activities, which are developed to strengthen the confidence to the customers, to the consumers, of the application of new technologies or new products. And this is conducted proactively in an initial phase of developing new system products, conducted proactively from industries, and I will present some data from the academia, but also from risk managers in the field of environment or in the human and animal health issues. So that's why we think it's timely to discuss, in this case, general issue arising with this particular technology we are dealing with. RNAi interference inherently has a high potential for being very targeted type of pesticide. This is because, as we know, there is a specific base pair complementarity which is needed to be targeted in the genome. And of course, it's very important that during the design of new double-strand RNA, we keep into consideration the genome of the target species. We have just heard some nice talk before, but also possibility of targeting unwanted, of course, but targeting other species or other pieces of the genome. So there are, in spite of this high selectivity, potentially there are some hazard there. We can basically discuss two types of these. One group that we can call sequence-dependent effects. and this one are, of course, if this gene, if our double-strand RNA targets, of course, a specific gene, but with a huge genome in insect, more than 10,000 genes, functional genes, it could be just by chance that there is another sequence that can be targeted by the double-strand RNA. Or the same target gene can be in the genome of non-target organisms, especially those that could be exposed in the agroecosystem. But we should consider also that there have been hypothesized some sequence-independent effects, the immune stimulation and the saturation of RNAi machinery. I will briefly touch upon on one final slide there. And if there is time, since it was also the topic of one question before, I have 1 minute to dedicate and just highlighting some issue of resistance developments. I will go through my presentation looking at this two aspects quickly, of course, these two aspects of any process that could lead to environmental risks. Let's talk about the exposure. We are today discussing RNA as a sprayable pesticide. So this is basically something that we will see when this will become available. So machines coming to the field and spray. So of course, the entry points of this pesticide in environmental systems are basically the sorry, I hear some noise in the background. Is somebody trying to speak? No, if I keep going. So I said the two entry points are the agricultural soil themselves, of course, including the plants, or adjacent surface of body waters that can be close to our cropped field. And this is, of course, the same for any exogenously applied pesticides. The fate depends on the application rate, machine handling operation, and so on. And well, we all know that extension services are spending a lot of efforts to increase the efficiency of the spraying procedure. So this already is a way to limit potential dispersal into the environment. Of course, these pesticides are not on the market yet. We have heard they will be pretty soon. So our information on the possible fate of this double-strand RNA in the field come from some experience with genetically modified crops. And it is foreseen that the amount of active compound that can be released in the environment, in the agricultural soils, are micrograms per hectare level. So we are talking about 3-4 order of magnitude lower than the cryoprotein release rates. And now there is a lot of progress done with the real-time PCR technique in detecting such small amount in the area. So this is good news from the environment. And it's not just that, it's a little amount possibly present in the environment, but we should remember that there is enzyme activity, microorganisms in the soil that can you know, start pretty quickly a biological degradation of RNAi, double-strand RNA in the environment. And there is a further limitation because part of these molecules could be adsorbed to solid water interfaces. As I said, since there is no experience with the pesticides themselves at the moment, of course, our colleagues in the industry may have already this data, but there is some similarity that we can use as a prediction coming from, you know, genomic DNA from plants and studies that followed their degradation in the soil and in the water. In particular, these two last paper I point to, this Doubleman et al. And Parker and Sanders have studied the persistence and the mechanism that lead to degradation in the soil. So there is some literature there. And I will present just a couple of slides with the main results from these two papers, which give a quite reassuring picture, because double-stranded RNA in the soil can last as an half-life of less than 1 day usually. This particular picture is degradation kinetics of the different dosage of RNA per gram of soil. And as we can see, independently of the initial concentration, after 24 hours, we have almost 15% remaining. So it's a rather quick degradation. A similar picture in the water. So if our focuses are the aquatic environments, this is an experiment done with samples taken in 2 freshwater bodies, one lake and one river, a simple experiment in which basically the sample was either left alone in a tube or first mixed and then left in this tube. And at the end, basically we have a half-life in the water, less than 3 days in both cases, but in the sediment it's even much less. So some remains in the water, but even there quite quickly degraded, not as quickly as in the soil, but quite quickly degraded. Of course, then we have to consider the exposure through the plant themselves, and basically it's the herbivory. And we know there is a huge number of course, I am an entomologist, so I used to make examples with insects, but other organisms as well. And not forget that there are some useful organisms. Well, useful is not a very nice word to use, but anyway, organisms that produce ecological services, such as the honeybee, that we have a picture here. It's also an herbivore. So there is a number of organisms which can be exposed to double-stranded RNA. And if we talk about pesticides, I think we can mostly forget about insects or organisms living in underground, because the spray is mainly affecting soil surface and plants. And we have clearly to focus our attention not just on the herbivore, but on the higher trophic level, where we have many species that provide ecological services in agriculture, natural enemies here, predator or parasitoid. So the question is, can double-stranded RNA move along trophic chains? And if it does, it is still active. One very nice study I keep quoting, because 8 years later, his first publication, I haven't seen no recent ones that are similar and nicely done to produce interesting data, interesting for at least 2 scopes. The first thing is, well, basically, I will describe quickly the exercise. You see with the arrows here, these small red spots. Whoever of you has experience with honeybees, beekeeping, knows that this is a serious pest. It's the varroa mite, a spider mite, which actually is an external predator of both larvae and adult bees. Well, in this experiment, the author of the study had the double-stranded RNA targeting the spider mite, so of course not the bees. And the idea was, if we feed the bees with the syrup containing the double-stranded RNA, can this, in this way, we reach control of the spider mites? This would demonstrate clearly both the transmittability along the trophic chain and the biological activity of the double-stranded RNA. And the answer is yes to both questions, because the experiment clearly showed that after a few days, spider mites started to die. So the double-stranded RNA was affecting the spider mites who were sucking, basically, the hemolymph of the honeybees. And also they were able to demonstrate the opposite. Once they had cleared this colony from spider mite, they used the infected spider mites were put on a new colony, so a colony who had no spider mites on it, and it was shown that the double-stranded RNA was carried back to this new strain of honeybee. So the trophic exposure is actual, and so this means that we could use double-stranded RNA to to control the population of spider mites through the bees, because the bees at the end of the experiment were clearly unaffected. However, this is not always the case, because we know that in several species, double-stranded RNA in the hemolymph is degraded. So in the honeybee, yes, but as I said before, 8 years later, I don't see many papers replicating this transmissibility on different trophic chains. I know several colleagues are working on this, including one small experiment in my lab. Even though the double-stranded RNA is acquired by the species, we have to figure out if there is a real possibility of physiological exposure, because there are many mechanisms that tend to inactivate the double-stranded RNA. First of all, how our molecule can enter a herbivore, an insect, whoever. Well, through the mouth, of course, if it is ingested, through the cuticle. We know that there are some part of the cuticle which are more permeable, and through the spiracle, which are the end point of the respiratory systems in insects. Now, I will not go in details, but there is this review. Well, it's a huge, it's a book, almost more than 100 pages, with which collected literature until 2017, and we know from this huge number of experiments that there is a large variability in efficacy. Well, the system was discovered firstly on Caenorhabditis elegans, this nematode, which is in which it's very efficient. It can be targeted by injection, soaking, feeding. It always works, but it's not always the case for other nematode species. Some of these animals have a different efficiency of silencing when ingested double-stranded RNA. When it comes to arthropod, we know for sure that our colleague have also presented nice data on Coleoptera, but also the first genetically modified crop expressing double-stranded RNA is also targeting Coleoptera, the westcorn rootworm, at very low doses. And however, among other orders, there is a large variability in efficiency. For instance, we know that Lepidoptera are less sensitive, generally speaking. Of course, there is variability within each order. Diptera are also less sensitive for oral delivery, even though we know that they can be targeted efficiently with injection. But the problem here is that there are physiological issue related to the uptake. And also in Demiptera, which is another important target as insect pests, that there is evidence for very large variability, even between different strains of the same species. So just to mention one of the mechanisms, there will be time next time to discuss more detail, but just to mention one, we know that, for instance, even though Lepidoptera do uptake double-stranded RNA, the problem is that in many of the experiments that exist in literature, these double-stranded are not processed to single interfering RNA, which are actually the active molecule. So even if Lepidoptera and now in this case, I think about non-target Lepidoptera like butterfly that may live nearby our cropped fields, even if those species as a larvae ingest some of this double-strand RNA, it is not given for granted that they can be harmed, because this lack of cleavage to single interfering RNA may not happen in some species. And well, we have heard, in fact, we have not heard, I understand colleagues could not give many details on type of formulation that we will find in the products that are almost ready to come to the market. I'm just mentioning some of the type of molecules that have been assayed in the literature. But the way I want to bring the point of formulation is that there is, in terms of environmental safety, there is a trade-off here, because I have just shown those slides that show that after, well, less than a couple of days, these are degraded on the soil, which is good. But of course, for a company who wants to have an effective pesticides, this environmental liability, it's a problem. So the formulation go in the opposite way. So that's why biosafety during the development of the product, again, I reiterate the concept, is fundamental. And now let's go to specificity, which is one of the most appealing features of this technology. This is one of the first paper from Wyatt et al., this paper which demonstrates specificity, because as I said before, non-target species may have the same gene, they may share this part of the genome with the target species. So this experiment demonstrates that even a double-strand RNA targeting the same species, in this case was ATPase specific, double-strand RNA was constructed for 4 different species. And here we can see in this table that the mortality, the high mortality was reached only when the 4 species, you can see in, on the x-axis here, the 4 species. And this high mortality was reached only when the species was assayed with the specific double-strand RNA for ATPase. And of course, there is a publication which was made by Pam Bachman and co-authors, and they work, well, at that time, I think was still Monsanto, it's Bayer nowadays, when they developed the maize resistant to the southern corn rootworm. They, of course, made a set of bioassays on non-target species chosen from different functional groups. And in these laboratory toxicity studies, you can see here what kind of measurement endpoint was taken, and there was quite a high specificity of their products. So in this case, bioassays were quite useful during the development of this new variety. However, a non-target effect may happen, and I will just show here a recent paper by Pan et al. It's 2020. This is, again, ATPase, double-strand RNA, were designed against the western corn rootworm, which is the target pest, but also was designed against 4 different ladybird species. As we all know, these are considered, you know, useful species in the agroecosystem because they feed on aphids, on other small herbivore. And when assayed with all kind of all different double-strand RNA, there was some effect, some significant effect, even though only at very high level of ingestion. And this effect, a negative effect, was quite different among the 4 coccinellid species. This means well, these are all coleoptera, so it's clear that there is a higher similarity in the genomes because the target species in this case is a coleopteran as well. The interesting thing here, I will just show you data which refer to the most sensitive, which was Coccinella septem punctata, and the least sensitive Harmonia xeridis. And we see here that against the two, let's say, non-target double-strand RNA, because this is just gas, so nothing in the genome of any insect, and of course this is water. We had the effect here, quite strong effect on one species, statistically significant effect on another species, but at much lower level. And the interesting issue here is also that these effects are paralleled by molecular analysis, and in which they demonstrated that the number of NMER matches, well, for those of you who are familiar with this application, from 2022 is the number which is considered to be useful for having quite active double-strand RNA. It's highest in Coccinella septem punctata. So this may likely be an effect due to sequence-dependent effect, because there is a similarity of this sensitive sequence. So this means that the role of bioinformatics may be important. The point is that at the moment we don't have such information on many non-target species, but we will discuss details in the next seminar. And another aspect is that these effects may also be unintended. This is another quite old experiment, by now 7 years ago. The experiment here was trying to feed honeybees again with some double-strand RNA, which has nothing to do with the genome of insect. Again, is against the green fluorescent protein here. There were a number of experiments done with the pre-pupae, larvae, and adults of the honeybees. And as we can see, we have a lot of genes that apparently at molecular level show some regulation, either upregulation or downregulation after ingestion. However, most of these are probably just a generic activation of some genes because of a strange diet, because when repeated, the experiment, only a few of these hundreds of genes were again downregulated or upregulated. Just to mention, as I said before, and there are hypotheses, at least for arthropods, of possible non-sequence related effect through immune stimulation or saturation of RNAi machinery. The immune stimulation, in fact, we know that there is a non-specific immune response to double-strand RNA in honeybees, in bumblebees. There is a recent paper. However, having a response to double-strand RNA, I want to point out, doesn't mean having an environmental risk. So we know that there is possibility that via activation of the immune system, something may happen. While for the saturation of RNAi machinery, this has been reported for mammals, but to the best of my knowledge, this has not been proven yet for invertebrates. The issue here would be this enormous additional amount of double-strand RNA molecule, because we know then it can be replicated, can be spread, etc. Can, theoretically speaking, block the RNAi machinery. If I have time, if I don't see Jeremy's face appearing, means that I have another minute, because I have mentioned then one issue, and there was a question before, is a possible spread of resistance. First of all, why in a biosafety framework we think about the insect resistance, insecticide resistance. We do that because clearly, it's mainly an agronomic problem, so that's why companies are working a lot on this. But it's possible from the environmental point of view, a possible drawback is that, in fact, if we have a pesticide which is quite environmentally benign, then if an onset of resistance appears, this means that we could go back to more harmful pesticide. That's why I think it's interesting that we look at this aspect in a biosafety framework. Well, double-stranded RNA, as any pesticides, with repeated exposure, with a higher amount, etc., they exert a selection pressure which can promote resistance. It's clear, and there is no chemical insecticide for which this cannot happen, and RNAi mechanism is not an exception. In 2018, Kajuri et al. Published the first report of a pest, again the western cone rootworm, which was resistant to a mechanism of gene silencing. It's a laboratory colony. This has not happened in the field yet. So this means that the mechanism is possible. And the thing here, and was mentioned before, I think, by the colleague from Syngenta, that one worry comes from the type of mechanisms, and this mechanism, which is based in this case on alter uptake of RNAi molecule. This means cross resistance basically to any other double-stranded RNA. At least in that experiment, that's what was found. So that's why the reply to the question was that no, different double-stranded RNA together probably is not the best strategy, but this will depend on system by system. And yeah, I think it's done. I haven't checked my timing. Hopefully I have time for a couple of questions. Thank you for listening.
Okay, thank you, Salvatore. Are there any specific questions to Salvatore? Oh, here we are, just, oh yes. What are the chances that double-stranded RNA can bind to clay particles which may lead to persistence in the soil?
Well, I will refer to the paper done by Kimberly Parker and her group. This has been proven. They can stay in the soil. The issue is both way, I would say, because on one side, they stay a little bit longer. On the other side, we might experience a limited bioavailability, because if this binding it's strong enough, this molecule will not be free in the liquid part of the soil, so cannot be absorbed and can hardly be eaten after all.
Okay, thanks very much. I think we still have Nina with us, and obviously Ethan. So I'm thinking that we can now have a more general discussion around some of the topics. There was I know this will come up in the second and third webinar, but I just wondered, particularly perhaps from Nina in Australia, if some of these biopesticides are developed, what sort of regulatory system approach will be taken? Will they be treated as another form of biopesticide, or will they be treated as a chemical pesticide, or do you think they're going to be considered as a novel category? I'll just unmute you. Yes.
Thank you. And as long as we don't put the video on, because it's late night now for me, so I'm sort of in a relaxed mode, but thank you so much. Really good to be still present. So in Australia, we have APVMA, which is the Australian Pesticide Veterinary Agency, which actually regulates the registration of these pesticides. We have had discussions with them, as a process to start this. The active, which is the double-stranded RNA, is likely to be considered as a biological active. So if we are going on the classification of pesticides based on the active, then it will be a biological active. However, if there is a carrier being used as well. Once again, these discussions are ongoing. My understanding, and this is just to say that it is my understanding at present, it's not a formal confirmation from anywhere, that it may be treated as a biopesticide or a biological control active agent.
And what about the fact that you're using bioclay nanoparticles? Does the fact that you're using a nanomaterial also complicate the regulation?
Yes, so the very good question. Once again, I think when we started with this, the selection of this particular clay particle LDH was really based on keeping the safety issues in mind, and also the definition of nano in mind as well. In most of the places, the definition of nano at present is anything between 1 to 100 nanometers. Since it's we have sort of progressed in formulations where we have addressed this aspect as well. This particular clay is really safe. It is also used as a slow release fertilizer already in agricultural applications. There is a lot of safety data available already on LDH, and we are looking into the size issue as well, so that we can, sort of quickly move into that space.
Okay, thanks very much, Nina. Ethan, obviously in the third webinar, we're going to have your colleague discussing regulatory issues in particular, I guess, in USA. But maybe you could just give us a brief overview of how the US regulator is viewing double-stranded RNA-based pesticides, particularly in relation to your product.
Yeah, so it's an interesting question. I think we would expect it to be registered as a biopesticide in the US. Of course, with it being the first sprayable RNA going through registration here, or we're expecting it to be the first sprayable RNA, that could change. So we'll see how it goes through, but we would expect it to be a biopesticide.
Yeah, okay, thanks very much. Salvatore, did you want to make any comment on this? Because you raised the question about the classification of this as a biopesticide or some other type of product. Oh, you're muted. Hang on, I'll just
No, I'm done that.
Right.
No, this I think would be a fundamental question, because on one side we know we are losing many molecules from the possibility for the farmers because of the new legislation in Europe. So having something that is, as we have seen, quite specific could confer quite confidence in from the environmental point of view. So it would be very important if organic producer, for instance, can use it or not. As well, in Europe, there is always a lot of discussion and trouble around this. So the issue here is that we have a purely biological process. So this molecule, even though they are, of course, but as any other organism that we use as microbial pesticides, for instance. So I think there is room here for having this introduced as a biopesticide, which would make quite a difference in my point of view.
Yeah, okay, thanks very much. I think we have rather run over time, and I think it's been quite a long session. You'll be pleased to hear that I think at the height of the webinar, we had around about 150 participants. A few people have had to leave and so on, but it's been very good. I'd like to take this opportunity once again to thank all of the speakers. It's been a very interesting session, a very good start to this series of webinars. I remind you that next Monday, we have I'll just put it up on the screen. Here we are. Next Monday, we have the next webinar starting a little bit later in the afternoon in Europe. And this is going to be looking very much more closely at some of the biosafety issues which Salvatore was referring to. So we'll be looking in depth at many of the issues which have been touched on today, and discussing these, because it's important to have an understanding of these so that we can then think about what information is required for assessing the safety of these biopesticides. And then I remind you that in the following week, the week after this, there will be the third webinar, which particularly will be looking at the regulatory aspects. Again, we have speakers from industry joining us, from Greenlight Biotech, and from Bayer, who will be talking about how they view the regulations, as well as Mike Mendelsohn from US EPA, and some European perspectives on this. So we look forward to you joining that webinar as well. So a reminder that next week we have this webinar, which is now hopefully on your screens. And then we have the third one, which will look into these aspects of whether it's a pesticide or not, and how it's going to be classified, and how the regulatory people are looking at it from different points of view. But it was useful just to get Nina's comment on that, because she's not participating in that third webinar. So it's good to get the Australian perspective on the regulatory issues. So once again, thank you to all the speakers. If you have any comments, Bruno and I would be very pleased to hear from you. If you have any comments about this webinar or about future programs that we should have, and also if you have any further questions or comments which you would like to go to the speakers today, please contact them either directly or send your comments and questions to Bruno and I. But we would like feedback from you, and this is the first time that I have been involved in organising a webinar. I think Bruno's a bit more experienced, but we're definitely on a learning curve, and any comments or contributions will be most helpful. So once again, thank you to all of you. I don't know whether, Bruno, you wish to just add a final comment.
I want to thank you, Jeremy, for the work that you did. It was really great for sharing this section. I hope that everything was okay. Yeah. From the technical point of view, like as Jeremy said, we will try to improve further. And I thank clearly the speakers and all participants. And as an average, we had 150 people connected. So I think that it's for the first webinar a good success. We will keep update you about the next seminars, and please, as you know, these webinars are open and free. So please disseminate the information, so maybe for the next we will be able to have even more people. So again, thank you very much and have a nice afternoon, and see you next time on the virtual webinar we are going to organize. Ciao.
Okay, thank you.
Thanks,
Ciao.
everyone.
Bye, everyone.
Bye-bye.
Thank you.
Hi, thank you.
Bye.
Thanks very much.